name is Julie Andersen. I'm a faculty member at the Buck Institute up in Nevada, California. For those of you who aren't familiar with us, the Buck Institute opened about 15 years ago. It's a freestanding nonprofit institute completely dedicated to study of aging and age-related disease. And what I'm going to talk to you today about is actually a joint effort between two laboratories at the Buck Institute. One of the laboratories, mine, involved in research in Parkinson's disease, and another laboratory, Judy Campisi's, whoops, and I moved it forward. Okay, sorry about that. Whose main area of interest has been in the study of a process called cellular senescence in cancer. And you would sort of think that maybe the two aren't very related, but I hope I can convince you with the data I'm going to present today that indeed they are. If you haven't visited the Buck Institute, you have to come up. It's a beautiful IMPA designed institute in Marin County, and really lovely just to see. So for those of us who study chronic sporadic disease, we tend to think of it as a triad that involves genetic factors, environment, and most importantly, for this particular conference, aging. So for example, in the case of cardiovascular disease, individual may be genetically prone for cardiovascular disease through particular isoforms of LDL receptors, et cetera. And then depending on the lifestyle of that person, diet, exercise, stress reduction, they may or may not manifest that. But of course, the most important risk factor for cardiovascular diseases is aging, and this is true of most of these chronic diseases. With cancer, there may be a genetic predisposition, which then, in conjunction with environmental factors, toxins that can cause a secondary hit, then you present with cancer. Incidence of cancers increase with age. So in terms of most of these chronic sporadic diseases, we're talking about this triad, and at the Buck, we're particularly interested in the aging aspect. So Parkinson's disease, like these other diseases, we tend to think of in terms of this triad, that there are some rare familial forms of the disease, but this only constitutes maybe 5 to 10%. The rest are sporadic and are likely due to this combination of genetic predisposition, environmental exposures over the lifespan, and that's something that my laboratory has been very interested in, and aging as the number one risk factor involved in these diseases. And in general, I think in the past, people like me who've been studying neurodegeneration tend to look at things sort of one disease at a time. But I think being at the Buck Institute has, over the last 14 years, has been a real revelation for me, because the people doing basic aging research at the Buck, like Judy Campisi, who's giving a talk in the next room simultaneously with my collaborator, realizing that aging may not just be a risk factor for these diseases, but could be an underlying driving force. And in terms of therapeutics, that treating some of these basic aging processes that are involved in the aging in humans, may act to prevent or alleviate these age-related conditions. And so the process that many of you may be familiar with, but which I will talk about today, is a process that Judy's laboratory has been heavily invested in studying over the last 20, 30 years. It's a process called cellular senescence. Cellular senescence is basically an anti-cancer mechanism in proliferating cell types. And the idea is that when you have a proliferating stimulus, this can drive cells towards tumorigenicity. And so evolutionarily, we've developed means of preventing that from happening. The most well-known process which can eliminate cancer cells is something called apoptosis. So it's basically a means of just removing those cancerous cells, taking them out of the system. Another process which Judy's lab has been on the forefront of studying is something called cellular senescence. Cellular senescence can be induced by a number of different processes that track with aging, oncogenes, telomerase erosion, DNA damage, and importantly, for the studies that we do in my lab, oxidative stress, because this appears to be a major factor involved in cell death associated with Parkinson's disease. And the idea that cellular senescence has developed because it protects cells early in life from cancer, but the issue is there is increased presence of senescent cells with aging in all organs, and unlike in apoptosis where the cells are removed, these cells persist. And there was a recent paper in Nature from Darren Baker at the Mayo where they showed in a progeroid mouse model that if they ablated these senescent cells, that it prevented a lot of aging phenotypes that were seen in these mouse, including cataract formation, fat deposition, etc., loss of fat deposition. So characteristics of a senescent cell, just like apoptotic cells, senescent cells are identified through particular characteristics. So of course we have the growth arrest, which occurs, which prevents proliferation of cells, but also you see features like loss of laminin B1 within the nuclear envelope, so reductions in this, work from Peter Adams and others, release of HMGB1, which traditionally is thought of something that interacts with DNA, but it also acts as an arrestin. When it's released from the nucleus into the cytoplasm and then secreted, it sort of sends out an alarm to neighboring cells that the cell is experiencing stress. Other phenotypes include particular types of DNA damage, foci, heterochromatin. A particularly well-studied phenomenon in terms of senescence is induction of a tumor suppressor called P16, and this is used as a biomarker for aging and senescence. And in addition to these effects, there's secretion of soluble factors, or something called the SASP, which I'll talk about in more detail as I go along. Okay, so while cellular senescence in the periphery is linked to several age-related pathologies, considerably less known about its potential role in the brain. That being said, senescent cells are cells showing phenotypes of senescence have been observed in the brain in relation to both normal aging, so both in humans and mice, and in disease. In Alzheimer's disease, a study by Bott et al. In 2012, and there are some unpublished data in terms of ALS. And we find, looking at in the affected regions in Parkinson's disease, which is primarily but not limited to the dopaminergic neurons in the midbrain in the area called the substantia nigra, we have found evidence of presence of senescent cells in these areas. The top panel here is looking at cortical astrocytes in Alzheimer's brain in this published paper showing presence of P16. I apologize for the quality of this picture, but in essence, when looking in PD brain in the affected region, the substantia nigra, we see presence of these astrocytes that show markers of senescence, including DNA damage, gamma H2AX, and also we see a substantial increase in levels of P16, this biomarker for senescence. I'm not going to show you an exhaustive list, but we've seen the thing about senescence, you cannot track senescence by a single marker. It has to be sort of this conglomerate of markers, and we see this in autopsy PD tissue. So if senescent, if non-neuronal, mitotically competent cells within the brain of PD or with aging are able to undergo senescence, so what? They're not proliferating. What damage can these cells do other than taking out those cells? Well, the concern is that these senescent cells, as they are maintained within the brain, can actually secrete compounds that contribute to age-related neurodegeneration. And this has been coined by Judy and others as a senescence-associated secretory phenotype, or SASP. And the SASP has been shown to be causative in things like cancer, and especially breast cancer paradigms. So we started out, as I said, very little known about the senescence process in the brain, and we started out asking the question whether senescent cells within the brain, whether they're present. So you have these non-proliferating neurons that ostensibly shouldn't be able to undergo proliferation in senescence, although there has been a report of that in older mice. Primarily, we believe senescence occurs in these dividing cell types in the brain, the most prominent of those being the astrocytes, but they could also include microglia, oligodendrocytes, et cetera. So are these senescent cells present? Do they produce a SASP, and what effect might that have on neighboring neurons? So the first paradigm that we looked at was an environmental paradigm, Paraquat exposure. Paraquat is an agent that elicits endogenous generation of cellular oxidative stress, which is something that we know elicits senescence in other cell types. Its systemic administration to mice is a well-established model of Parkinson's disease. There's extensive epidemiological data linking lifetime Paraquat exposure to risk for Parkinson's disease. In fact, of all of the pesticides and herbicide studies, probably Paraquat shows the strongest link in terms of being a risk factor for the disease. Also, recent data suggests that genetics may play a role, that genetic susceptibility, presence of particular alleles involved in glutathione metabolism, et cetera, can enhance susceptibility to Paraquat in humans. So the first thing that we did with Judy's lab was to look at the effect of Paraquat in culture on human astrocytes. The reason we chose astrocytes is because they're the most prevalent cell within the brain. It's not that we think this only occurs in astrocytes, but this was kind of a starting point. Oh dear. Okay, well, you'll have to just believe this. This is probably a Mac to PC conversion. So this is my, I don't know, foreign version of the slide, but I'll go through it with you, and you'll just have to believe me. So over in this corner, we're looking at SA beta gal staining with increasing concentrations of Paraquat, which is quantitated below. And we can see that with increased Paraquat, we get increase in this blue beta-galactosidase staining. This senescence associated occurs at a particular pH. And again, this is a hallmark for senescence. So we treat these human astrocytes with Paraquat, and we see this increase in SA beta gal. Lower panel here, we're looking plus and minus Paraquat, and we're looking at the nuclei and presence of these DNA damage foci. We see this increased with Paraquat. We see an increase in P16 expression in these cells, and we look at, wow, and they've even got exclamation points at the end. Okay, so and we see an increase of this sort of prototypical SASP marker IL-6. So we see presence of both senescence after Paraquat induction and of SASP. Right, okay, I used the wrong, okay. So it does seem that we can elicit senescence within these human astrocytes via Paraquat induction. So what effect might this have on neighboring neurons? Well, if we take conditioned medium from these cells and we put them onto either, on the side over here, dopaminergic neurons that have been derived from iPSC cells, or we look at neural stem cells, we can see that there's a significant increase in toxicity in the Paraquat-treated cells. If you look at BRDU labeling in the NSCs, we see a reduction in proliferation with increasing Paraquat levels, and we see decreased migration, which it's not clear whether that's due to a loss of proliferation or a migration, but we do see on the scratch test that Paraquat inhibits closing of the scratch. Okay, so what about Paraquat administration in vivo? And most importantly, because all of the studies up until now in terms of neurodegenerative disease in humans have been correlative, does senescent cell ablation prevent characteristic neuropathological phenotypes associated with this model? And for these purposes, we used a transgenic mouse, which had been constructed by the Campisi lab, in which they can ablate senescent cells. So in essence, they're driving expression of a couple of markers behind the P16 promoter. So in cells that become senescent, they will be displaying both luciferase and RFP. And they also have a viral HSV-TK, which when you feed these animals ganciclovir or you inject ganciclovir, then it results in a phosphorylation, DNA chain termination within mitochondrial DNA, and it ablates the cells. So in essence, in this construct, you can both look at what cells become senescent, and then you can ablate them and see the effects of that. And in looking at various markers, well-established markers for senescence within the periphery, we see that those markers are also impacted in this Parkinson's disease model and are prevented when you ablate the cells. So for example, nuclear HMGB1 levels are found to be reduced in the presence of Paraquat, which is a mark of senescent cells, and they are prevented by ganciclovir treatment. And I just want to make a point that this data, sort of midway through my talk, this data is actually part of a poster that's being presented by a postdoctoral fellow who's sitting in the back. And if you want to have a more detailed discussion about this, please visit her poster. She did a beautiful job. It's a beautiful poster. Please go by and talk to her. Georgia Wood. Okay, and she did this lovely, lovely work, which took hours and hours poring over the microscope. So it's always funny to show one slide of something that was very intensive research. Looking at nuclear lamin B1 levels, again, we see that, and I should mention these are in astrocytic cells, and we're also in the process of looking at other mitotic cell types in the brain, but we're looking at astrocytes here. We see a reduction of this characteristic marker, the nuclear lamin B1 levels. They're reduced in the astrocytic cells, and we can remove them with ganciclovir by ablation. And most importantly, removal of these cells actually reverses the neurodegenerative effects that we see in this particular neurotoxin model of Parkinson's disease. So looking at A, we can see that we see an increase after Paraquat treatment in markers for both senescence P16 and the SASP IL-6, and that they are removed when we ablate the senescent cells, as you would expect. If you look at dopaminergic cell loss in this particular Paraquat paradigm, which we expect about a 30% cell loss of dopaminergic cells within the substantia nigra. This has been shown by several groups. This is prevented by ablating the senescent cells and looking at motor function, which is known to be down in Paraquat treatment. This is reversed when the senescent cells are ablated. This is in the cylinder rearing test. And when we look at things like adult neurodegeneration, so this concept that neurons in case of injury can re-enter mitosis and replace the lost neurons, this is known to be down following Paraquat treatment, and we can restore this by ablating the senescent cells. Okay, so in this first paradigm, we were looking at an environmental factor that tracks with Parkinson's disease. However, it's in young animals and it's acute. We were very interested in what would happen in a more chronic age-related models. And the model we decided to use is a model that had been constructed and characterized by our lab, where we're mimicking the age-related changes in the mammalian brain via the induction of an enzyme known as monoamine oxidase B, or MAOB. So MAOB is traditionally found in the astrocytes. It increases with age in the brain. It's also associated with age-related neuropathologies, including Parkinson's and Alzheimer's disease. And actually, MAOB inhibitors like deprenyl and rasagiline are currently used as clinical therapies for the disorders. And the system we set up was a DOX-inducible system where we can, by adding doxycycline to the feed, using an astrocytic GFAP promoter, we can drive expression of MAOB, and we've published several studies showing that this results with age, by 12 months of age, in a Parkinson's-like phenotype. Looking at markers of senescence within these cells, what's shown at the far panel at the top is beta-gal staining in 3-month-old animals. The next panel is 12-month-old animals without any treatment, and you can see an increase in the beta-gal staining. So with aging, by 12 months of age, we see an increase in presence of senescent cells within the mouse brain, which is intensified with oxidative stress elicited in the astrocytes by MAOB. And looking at, looking at P16 in non-treated and treated, or non-dox and dox, we see an increase in P16. We see an increase in IL-6, which is a marker of the SASP. And when we look at the impact of ablation of these cells, I think the top two are the markers. So this is looking at luciferase goes up, is ablated is decreased by senescent cell ablation. The second panel at the top is looking at the RFP, and the bottom panel is looking at the P16 RT-PCR. So in essence, in the second age-related chronic model, we're also able to induce senescence and prevent it through cell ablation. What are the effects in terms of neuropathology in this model? We've also extensively published in this model that with dox induction at 12 months of age, we see a 30 to 40-fold reduction in S and dopaminergic cells, which we can reverse to a large degree by senescent cell ablation. And this also has an impact on motor behavior. If we look by open field analysis at either ambulatory distance or velocity, we can see that it's reduced, and we've shown this before in this model, and restored by senescent cell ablation. So these data suggest that cellular senescence is likely an important contributing factor associated with brain aging, and it could constitute a novel therapeutic target, not just for Parkinson's, but for other diseases and for aging. So we have a long way to go with this. Future directions that we're heading in is senescence induced by Paraquat or other environmental toxins. We assume it's a result of oxidative stress, but we're looking at this. We have a grant through NIEHS to look at the ability of other environmental toxins to induce senescence within the non-neuronal cells and to impact on neurons. So we're doing a screen to look at other factors that have been associated environmentally with Parkinson's disease. And probably most importantly, we think these effects are non-cell autonomous. We don't think it's direct effects on the neurons, although, as I said, there's a single publication suggesting that the neurons can undergo senescence. Are these non-autonomous effects elicited due to direct effects of the SASP on the neurons, or are they via activation of neighboring glial cells, frank activation that can affect neuronal cell survival? So in other words, SASP being produced by, for example, astrocytes, then eliciting microglial activation that can have an effect. And if you think about the various cell types, or the various non-neuronal cell types within the brain, you can come up with many ways by which, if those cells undergo senescence, they could be impacting on brain function. And that's really what we want to gain a better understanding of in our studies. And I think that's it. I will stop and entertain questions.